the topic that we have for today is enzyme. Let's talk about enzyme in detail. That what is an enzyme? So enzymes are basically biological catalysts that speeds up a chemical reaction. So if in a reaction enzymes are involved, that reaction is basically speeded up and it is performed very rapidly. So enzyme contains some substances like let's make a structure of an enzyme. For example, this is an enzyme. All right, so let's name it as enzyme and let's have a look at its structure. This is a groove, or you can say a depression on an enzyme. And what is meant by this is basically an active site. The active site of an enzyme is a site where a substrate is attached. So for example, we say these two are the substrate that are going to be attached onto the active site of an enzyme in order to form a product. So what is a substrate? So these two are substrate. Substrate can be one, substrate can be two, they can be multiple in numbers. However, the product is always supposed to be one. This is something that we need to remember. Then what we need to know is that what is an enzyme substrate complex. So when a substrate is attached on the active site of an enzyme, it forms a structure like this. This means that substrate is attached onto the enzyme. Do remember that substrates are always specific because they cannot be attached to any kind of an enzyme. They always be very specific when it comes to an enzyme. So when the enzyme and the substrate are binded together, they are called as enzyme-substrate complex. Once the enzyme-substrate complex is formed, we say that this forms up a product. Okay, so we already discussed it before that what is an enzyme. Enzymes are the biological catalysts that speed up a chemical reaction. And what is a substrate? Substrate is a substance that is attached on the active site of an enzyme and forms a substrate complex, that is an enzyme substrate complex, and that in turn forms a product. So one thing that we need to remember is that these substrates are always specific in nature. It is not attached to any kind of an enzyme. For suppose, let's take a look at an example that says starch is converted into maltose in the presence of an enzyme, for example, that says amylase. Okay, so we need to know that this is a substrate. Okay, this is an enzyme. So definitely some kind of specificity must be among the substrate and an enzyme that amylase has acted upon starch to convert it into maltose. Okay, so let's have a look on some graphs. Let me explain with the help of some graphs. All right. We're going to discuss graphs in detail. That, for example, says we have this kind of a graph that have a long curve. So this side of a graph shows us the progress of a reaction. All right, and this side shows us the energy. All right, what does this huge curve shows us? This shows us the activation energy. Now comes up the question that what is meant by the activation energy. Basically, the activation energy is the energy that is required to start up a chemical reaction. Alright, so this is an large activation energy that is required by a reaction to be proceeded. Alright, and some basic that we need to know, that here we have the reactant and here we have the product. Let's look at another example, and then I'll explain you what is the difference between the two graphs. So this graph have a slight curve right over here. That means the activation energy here is low, and the activation energy in this graph is pretty high. Why is it so? Because the enzyme in this graph is not present. Enzyme is not present, whereas in this graph enzyme is present. What does the enzyme do to activation energy? It basically lowers the activation energy so the reaction can proceed faster. Alright, so this is the difference between the two graphs. Let's give it a review. That this is a graph without an enzyme. That means the activation energy is very much boosted. Reactants are being converted into the product, and due to the high activation energy, reaction is very slow. Whereas in this reaction, or in this graph, you can say the enzyme is present. That's why the activation energy is very low, as we can see by the small curve, and the reaction is proceeded very faster.